go. All right, so thanks everybody for coming today to our RCN-ECS happy hour and panel on artificial breeding and reintroduction. And this is accompanying a couple of readings that we've done in our RCN-ECS spring reading group on this topic. So really excited to have some super interesting panelists who do super interesting work here to talk about their research. So I'm going to just go through and give a short introduction to each panelist, and then you can give a summary of your work. Just talk about how it just generally what your research focus is, your various research topics, but also kind of how that kind of touches on artificial breeding or reintroduction or things like that. So all right, great. We're gonna start out first with Evelyn Jensen. So Evie is a lecturer in molecular ecology at Newcastle University. And she was an author on one of the papers that we read, which was the temporal monitoring of the Floriana Island Galapagos giant tortoise captive breeding program. So, Evie, do you want to give a kind of brief summary of your research?
Yeah, absolutely. Well, thanks for inviting me and organizing this. I think it's going to be fun. So I think my research that's sort of most relevant to this topic really is relating to the Galapagos giant tortoises. For more than 10 years since my PhD, I've been studying the evolution and conservation of Galapagos tortoises. And in addition to being sort of charismatic emblems of evolutionary biology, they are also really outstanding conservation success stories. And that's largely due to some very successful captive breeding as well as head starting programs. And I've been involved in sort of the genetic planning and also the genetic monitoring of these types of programs. So the sort of most exciting, of course, is of the Floriana species, since it is considered extinct, but lives on in these hybrid individuals. So yeah, thank you.
Cool. All right. Thank you. Yeah, next we've got, I'm going to co-introduce Rebekah Horn and Shawn Narum. So both Rebekah and Shawn work at the Columbia River Intertribal Fish Commission. Rebekah is a fisheries geneticist and Shawn is the lead geneticist. And Rebekah and Shawn were authors on one of the other papers that we read. So the paper on detecting genomic variation underlying phenotypic characteristics of reintroduced coho salmon. So yeah, Rebekah and then Shawn, if you guys want to introduce yourselves briefly.
Sure. So I'm Rebekah Horn. I've been working with, we call it CRITFIC is the acronym, for about 5 years as a geneticist. So my focus is mostly on species of salmon, Chinook, and coho. And we're using sort of population and conservation genetic metrics to help reintroduce and as well as manage populations throughout the Columbia River Basin. We work for a group of 4 Columbia River Treaty tribes. And so everything that we do is basically for them and basically how we can assist with genetic methods in helping to sort of any sort of questions they have about salmonids. So the paper that Shawn and I wrote a couple years ago focused on coho salmon. It's in the upper Columbia or mid to upper Columbia River within Washington. This is part of the Yakama Nation ceded territory. They coho were sort of extirpated there in, I think it was the mid 2000s. And so they wanted to bring coho back to their original sort of watershed. And so they've been taking downriver stocks and stocking them into the Wenatchee River, where we focus this particular paper. And we've been helping to using genetic methods to monitor that stock since about 2012. Now that they have a returning stock to that location, they also are interested in sort of phenotypic variability that can exist naturally, and how do we introduce that? And so that's what the paper was trying to get after, is if there has been any sort of genomic regions of selection that have arisen since these initial introduction in the population. And then I'll let Shawn sort of take over, introduce.
Hi, everyone. Thanks for having us today. It's fun to talk to this group about some of the work that we do. As Rebekah indicated, most of our work is focused on conservation genetics, genomics approaches to support salmon and other native fish recoveries to support the Native American tribes in our region. And I guess a lot of the work that we do is, due to the reality that there's been dramatic loss of these native fishes in the Pacific Northwest, particularly in the Columbia River. So today we see about 5% of the abundance that we used to see, particularly when the tribes signed some of their agreements in 1855 with the federal government that would allow them access to these routine fishing sites. That was a priority for them. And so now what they're facing is a very dramatically different situation than what they had signed those agreements back, over 150 years ago. So there's concerns about how we mitigate for the loss of those salmon. And as Rebekah mentioned, it's coho salmon. We do a lot of work with Chinook salmon and steelhead, sockeye salmon, but we also do some non-salmonids that are also particularly important to the tribes. Pacific lamprey are some consider much less charismatic species, but are also critical to these tribal cultures. And we've done a lot of work in, particularly there, we're using a lot of our genetic techniques for translocation efforts, where they're restoring them to places where they more used to typically occur. And we're using some genetic pedigree methods to track the success of those translocation efforts and things like that. We've also done a lot of work in white sturgeon recently, much more complicated genomic species because they're octoploids. But anyway, I'm glad to be here today. Happy to talk about some of the artificial breeding that we do. Rebekah is definitely the one that can dive into the details on the coho salmon, but also happy to provide some insight towards some of the work we've done on other species. All right, thanks.
Great, thank you, Rebekah and Shawn. Okay, so next we've got Jared Westbrook, who is Director of Science at the American Chestnut Foundation, so moving to plants as well. So Jared, if you can give a quick summary of what you guys do there.
Hey everyone, it's great to be here today. And I work for the American Chestnut Foundation. It's a nonprofit based in Asheville, North Carolina. And we've been working with breeding. Well, the story of the American chestnut is a blight was introduced, a fungal blight from Asia, in the turn of the 20th century. So 1904 in New York City, they found these trees that were infected, and then it spread across the entire range of the species and killed billions of trees. So in Asheville, North Carolina was in the mountains, these trees grow on top of the mountains. Yeah, they're just little sprouts right now. And so there's been this effort, kind of a grassroots effort since the 1980s. I mean, really, even from before then, in the 20s, the 1920s and things, people are trying to breed the Asian chestnut species with American chestnuts that occasionally they flower, they'll get the blight, or they flower and then they get the blight and they die back. So we can breed with the Asian species. So we've been doing that for a long time, and have learned that we initially had thought that the resistance was pretty genetically simple, but it's actually really complex. And so we've been learning oh, this is quantitative resistance we're dealing with. We were trying to do back crossing to get that resistance in, but as we do that, we dilute out some of that resistance. So we also have a transgenic tree. It's one of the first examples of a genetically modified tree used for conservation. And this gene that we put in comes from wheat, and it detoxifies oxalic acid that's produced by this fungus. And it keeps the trees are able to tolerate living with the blight. So that's going through federal regulatory review right now, and we may have approval to release these trees as early as this summer. And if we can do that, then our next task is to introgress that gene into the wild population, a representative sample, and develop seed orchards that we can, start repopulating the forest with these resistant trees. So yeah, that's it.
Awesome. Thank you. Thank you, Jared. Okay, and then our last but not least panelist is John Parkinson. So John is an assistant professor at the University of South Florida, and he was the author of, I think, one of our optional readings, which was Molecular Tools for Coral Reef Restoration Beyond Biomarker Discovery. So John, can you give a little intro to your work?
Sure, thanks. Thank you all for having me and for attending at all the different time zones. I appreciate that. Yeah, so I think the most relevant work that I do for this discussion relates to the time I spend with the Coral Restoration Consortium, which is a group of scientists put together by NOAA to kind of provide guidelines for ongoing coral restoration efforts. So yeah, we're dealing with an invert, we're dealing with the marine system, and we're dealing with a pretty existential threat in the form of climate change, right? And so there's a lot of interest in potentially breeding for or selecting for more thermally tolerant coral holobionts. So the whole organism, not just the coral itself, but its microbial partners, including symbiotic algae, which is what I usually actually do research on. I'm really focused on algal genomics and their systematics and taxonomy and how they impart heat tolerance to hosts or not. But through this Coral Restoration Consortium group, we've put out a couple of these sort of guideline publications. And we have one in review right now specifically on assisted breeding. So I don't think I'm an expert by any stretch, but I've talked with a bunch of people who are. And so there might be some interesting tidbits to take from there, because the marine realm is pretty different, I think, from terrestrial, and yet there are also a lot of shared commonalities. I really appreciate the work that everybody else has been doing, because it's helping to inform what we're doing with corals to the extent that we can. So yeah, thank you.
Great, thank you, John. Okay, I just want one final introduction of someone who's not here, but who is super important to putting this panel together. Melissa Noggle got in touch with all of the panelists and gave me a little outline for some questions, which are really great to get things started here. She's unfortunate. She's doing a PhD at Southern Cross University in Australia, which is why she's not here. So she's got some pretty serious time conflicts. But yeah, we would not be having this panel without her. So important to give her some recognition for that. Yeah. Okay. Well, thank you, everyone, for coming today, and I'll start off with some just kind of some questions to get things started for the panel. I guess one question, thinking about, some of us who don't do work with artificial breeding, I think a lot of us are working more with wild populations, but I think we use a similar set of tools, a lot of genomic tools, a lot of maybe reduced representation or whole genome sequencing. So I just was wondering, wanted to ask you guys, what kind of tools are especially useful for you in doing artificial breeding and in doing restoration, and how do these tools differ, when working with these captive populations or kind of artificially breeding populations compared to wild populations?
Do we have a conch shell that we should pass around?
I think you can start if you would like, but yeah, I think you can raise your hand too if you want to add something.
I guess I'll stick with just a simple one that is sometimes overlooked in the context of all of the new great shallow genome resequencing we can do, we still use microsatellites quite a bit, at least in corals. They're clonal, right? And in these nurseries where we're trying to grow them, we have to keep track of who's who. And you cannot do that by simply looking at them, right? And even our efforts at tagging them, again, once you get into the water, it can actually get very confusing, even laying out our nurseries and who goes where. And so you periodically have to retest them. And Ileana, my former advisor, recently came out with a SNP chip that has been helpful for providing a higher resolution for basically the whole collection of Caribbean acroporid corals that she's had or people have sent her. But she's basically aligning that with the microsatellite data that was already out there, and for the most part, it lines up well. Obviously, you pick up on some genets that you didn't necessarily detect the first time, but for a quick and dirty, again, in our restoration context, we're trying to come up with simple or relatively inexpensive or easily accessible tools. And microsatellites actually still work well for some basic tracking of genets and then associating phenotypes with genotypes.
In the chestnut world, I've spent a lot of time working, like building up genomic resources. So we have, reference genome for the American and Chinese chestnuts. And in the applied breeding sense, we've done a lot of phenotyping on pretty large populations, thousands of hybrids between Chinese chestnut and American chestnut. And so, we've genotyped them with GBS, and I can predict blight resistance from that GBS data, like associating genotype with phenotype. And I think our goal is to get trees that are, retain some larger effect alleles, which there's maybe a few of those, and also have most of their genome inherited from American chestnut, so we can just look at local ancestry across the genome, using that GBS data and markers that are, fixed differences between the species. Another thing we've been doing is sequencing the whole genomes of a lot of wild trees from across the range, and seeing, doing genome environment associations, and seeing what's the adaptive allele frequencies in each of the subpopulations, and seeing how many trees would we need to breed with to conserve, let's say, to match allele frequencies at, let's say, R squared of 9.99 or 9.5. And it's not, there's millions of these trees out there, and we only need a few hundred to actually breed with to represent the diversity from the southern, middle, and northern parts of the range. So that's been really useful. And the way we speed up the breeding, so we're talking about trees, short generation times or long generation times, is we grow the seedlings under 16-hour long day grow lights, and they'll produce pollen in their first year. And then we can take that pollen and put it onto additional wild trees. So that's how we sort of introgress into this transgene into a wild population. We can genotype those selected, or the progeny from those crosses, and select out the transgene founder genome, the founder genome that we use to insert the gene, and select 4 more of the wild diversity. So we're using genomics in a lot of different ways.
Yeah, I can add in that in our group, we use SNP panels for the majority of our monitoring of the different salmonid species. And we do that in a genotyping by sequencing approach, or GT-seq, and that's very cost effective in genotyping, because we genotype, it's over 100,000 fish every year in our lab, just hatchery fish for tracking via parentage. And for coho, there's only 250 something SNPs within the coho panel, and that's enough power for us to be able to detect parent offspring pairs very easily. And like I said, it's quick, it's cheap and effective. So that's sort of our main go-to for genomic data. And then we're using low coverage whole genome resequencing data, also to get at some of these questions about adaptation and how we can use that to factor into management. And that's definitely facilitated by the fact that there's high quality genomes available for most of the salmonid species. So that helps a lot. But we're using the whole genome resequencing that we do. It's sort of outlined in the Coho paper that we individually barcode individuals, and everything gets then pooled for sequencing. And that again allows us to be a little bit more cost effective on the number of individuals and populations that we can resequence. So that's what we're using.
And I'll just chime in with a follow-up comment there. And I think one of the questions was, what is the differences between tools that we'd apply for captive populations versus wild populations? And really, we study both, and that's because they're co-occurring in a lot of the rivers that we're studying. But I would say it really depends a lot on some of the questions, like Rebekah was just pointing out. If we're trying to develop pedigree relationships with some of the genetic techniques, low density panels have been very effective for that. I like what John said about microsatellites still have a very useful purpose for parentage type of applications, even though a lot of us have moved away from them. They still have a lot of utility. Some of these low density SNP panels that Rebekah mentioned are very cost effective for us for doing these pedigree relationships because we're genotyping these vast numbers of fish. But we also have lots of questions trying to understand local adaptation to of these species to local environments. And so that's where some of these genome-wide or whole genome scan approaches are really more effective to help us understand the adaptive regions of the genome and detect those more effectively. And I will say that we have shifted much more towards the whole genome sequencing and away from something like a reduced representation. If we're trying to detect adaptive regions, we just have much more power with that higher marker density, much more confidence in those adaptive regions when we do detect them. I will say we had some success with the reduced representation approach when we've tried it, but when you have one marker on a chromosome that shows significance versus 500 that are tightly associated in this nice peak of LD, the level of confidence there is quite a bit different and quite more robust as far as making some inferences about that adaptive region. And anyways, I think this is a topic we could get stuck on for a while, just because it's honestly one that I've presented to a congen workshop that happens every year through University of Montana, and we spent a couple hours talking about this with different issues. But I like the discussion. I'll just go ahead and wrap it up there because I think there's a lot of things that we want to talk about too.
Anyone else have any Evie, do you have any thoughts on this?
I think maybe my perspective is both very similar and then just a little bit different. So for the tortoises, we have been using microsatellites. We're just now sort of transitioning to where we've now got whole genomes for all of our captive breeders. And we're using that to try and disentangle ancestry. We know they're hybrids. The difficulty comes from not having references since one of the species is extinct. There's very few museum specimens. They have terrible quality DNA. So that's a whole other tangent. But sort of looking forward, we'd like to do a GTC panel to be able to assign parentage, that kind of thing. But we're kind of stuck with this issue of coming up with markers that we can employ in the Galapagos, because we'd like to be doing a lot less exporting of samples and opportunities. So what are the sort of genomic markers that you can deploy, with very basic lab facilities, that kind of thing? I don't have the answer for it, but that's actually sort of more of our future concern than, can we get fancy genomic markers? Can we do this in location?
Very cool. And I also saw, so Jared also had a question in the chat on, I guess for anyone who's working with, thousands or tens of thousands of individuals as you are in some of these breeding programs. So how do you scale up the DNA extractions? Or how do you keep track of all of those?
I was just responding in the chat to Jared. I think that's a good question of, these DNA extractions are certainly a slow part of the process. And for our lab, we've had to take the approach over the years of hiring full-time lab technicians. We have 7 to 8 at any given time throughout the year. And there's, that's not the only part of their job, but that's one of the roles as far as prepping these libraries, extracting DNA. And it's just one of those things that we've tried to find a way to automate this in the past. And every time we try to get our lab techs to use the automation that we purchased, they very quickly abandon it and go back to doing it manual, because they can do it more effectively manually, even though there might be some time efficiency with the robotics. It usually ends up being a bit more challenging to use some of those robotics that we've invested in. And so we have actually some pieces of equipment that were pretty expensive that are just sitting idle on our lab benches, because our techs are much more efficient at really just maintaining a well-organized system to track the samples, get everything set up in a way that allows them to process them in a kind of a routine protocol throughout the week as they go through these large numbers of samples. Brute force, I guess, is the answer.
All right. Anyone else have any insights on that? Is it very different for corals, or?
We tend not to have tens of thousands of samples that need to get processed annually. Smaller population, at least when it comes to the acroporids. Yeah, it's definitely a bottleneck, and that was why Ileana spent so much time investing in developing the SNP chip. With the other added benefit that apparently it can be read off of machines that are commonly found in hospitals. They typically have them there for diagnostics. And so it was another idea towards having local individuals do the local work where possible, rather than having the bottleneck of sending samples to, basically at the time it was just one lab. As coral restoration has expanded pretty much exponentially over the last 10 years or so, that things have shifted. And so some of my memories from that dark time. But yeah, these days, DNA extractions are still something that we don't have a fancy robot, alas, but I'm glad to hear that we might not necessarily need one.
Okay, great. I had actually this question is maybe primarily to you, John, at least first. But what are biomarkers? When are they useful, and how do you validate them?
Okay, so biomarkers are just indicators of a process, right? We tend to split them into the category of diagnostic markers versus predictive markers, right? I often give the example of blood pressure as a biomarker, right? You go in, you get it checked, and if you're having a heart attack in that moment, your blood pressure could potentially tell you that. But also, more on the predictive sense, you monitor that because hypertension over time is something you can track and predict whether or not you might have heart disease down the line, right? And so you can use that information to anticipate how something might respond biologically in the future. And in the coral realm, we really are interested in both. A lot of the more traditional biomarkers that have been used in the past or have been used regularly are diagnostic. They tell you that, yes, there was perhaps a pollution event that took place nearby recently. We have a technique for measuring photosynthesis, right? Pamphlorametry. You can look at the algal photosynthesis and use that as a biomarker for current stress, because as the symbionts get stressed, they basically photosynthesize less, and you can detect that and use that for a more immediate sense of, yes, this coral is potentially healthy in the moment, or it's experiencing stress. Color is another indicator. We're hopefully familiar with coral bleaching, right? As the corals get stressed and they lose symbionts, you can detect that visually. They can lose a lot of symbionts before you detect it visually, though. And sometimes bleaching, just because you've lost a lot of symbionts, doesn't necessarily mean that you're unhealthy in that moment. It could be part of a natural sort of regulatory process. So all of these indicators have pluses and minuses, right? And that paper that you guys optionally read had sort of a breakdown of different potential biomarkers that have been tried in the past, or we think they might be useful in the future, particularly now as we're trying to predict and anticipate which corals might survive, and which ones might have traits that would be useful to, say, propagate throughout the population. Something that's a bit unique to corals, maybe, I don't know, is that they're kind of hard to phenotype. We don't have many very good measures or metrics for how healthy they are or how they're performing. We have a couple that we've used in the past that we thought were good, but then turned out to be not so great. Growth rate, for example, is something that's incredibly variable in corals. And just because you're growing slowly doesn't mean you're unhealthy. It could have everything to do with the local conditions, water flow, things like that. So while growth rate in a linear direction is really helpful, if you look at, say, calcification rate, that seems to be very genetically determined. And so there are these different processes that have different utilities for predicting how things might perform in the future, whether they're actually tied to genetics strongly or if it's a more environmental trait. So a lot of the biomarkers talked about in that paper are sort of molecular biomarkers looking at gene expression or protein abundance, and trying to suss out from that whether that's reflective down the line of, say, disease resistance or heat tolerance. And so that's where validation comes in. That's the whole point of that paper, is you can discover a lot of biomarkers fairly easily because we have an overwhelming amount of data, or I'll call them potential biomarkers, right? But whether they actually reflect the process that you want and whether you can actually use that trait in particular to anticipate future outcomes, that's really hard to do on the time scale that we have, because we're really talking about like decades, right? In a couple of decades, most of the corals might be gone. So how do we very quickly determine that this marker is useful and something that we can scale up and actually use and have practitioners use when they're managing nurseries or managing coral reef ecosystems on the whole? And that's where the word of caution came in, that, you know, we don't want to promise the world with these things. It's a tool. If we had a bunch of time, I'm sure we could develop a lot of useful tools. There are some biomarkers that seem really promising in corals. Someone did a study and basically was able to classify whether a coral was going to be resistant to a disease with like 90% accuracy. And that validation step involved taking some corals and exposing them to disease, and, you know, checking to see if they had the biomarker before they were exposed or not, or whether they were, you know, plus or minus for that particular molecular marker. And so it can be good, but we don't want to overstate it, because as far as I know, even though that marker had really good resolving power, it's never really been employed en masse. And that's the issue with a lot of these biomarkers. It's the practical side of actually being able to use them regularly. And again, for corals out in the water, it's a challenge.
Great. That I think that leads into kind of another question more generally. So how do you well, one, how do you kind of choose your brood stock, or how do you kind of get that out of the wild and select what you need? Have any of the panelists have any, you know, challenges in that you've had to overcome when selecting brood stock? Are there are you worried about adaptation to captivity when you're kind of doing artificial breeding and selecting brood stock? And, you know, what are all the considerations involved in that?
I can start specifically in coho salmon, because I mentioned that they're completely extirpated in a lot of the sort of mid Columbia and up, like into the Snake River Basin and stuff. So they weren't around. So there wasn't really a stock to pull from. And so they went to some of the hatcheries along the coast and pulled stock from there. And then it was just sort of a matter of wait and see, as if we release, you know, offspring in these upriver regions where they weren't born, will they return, you know, in a few years? Because that's the life cycle of all these salmonid, you know, born in freshwater, go out to the ocean for a year or so and come back to where they were born. And it turned out, you know, these lower river stocks actually did pretty well within just a few years, like a generation or so of coming back frequently. And now we've got, you know, coho that are returning naturally to these systems in really large numbers, actually. And that sort of not only the program that was in the paper, but there's several programs, all pretty much led by tribal programs that are returning coho in pretty large numbers. Now that's definitely not the same for Chinook and steelhead in the Columbia River. That definitely depends on, if the stock has been extirpated, where's maybe the next closely related genetic stock that we could pull from. Maybe there are a handful of wild fish that come back to that region, and could we start a broodstock based on those? And then there's, of course, the questions of, are you going to be, some sort of bottleneck effects or founder effects in those type of populations? And that's always a big question for salmon hatcheries in general, is how do you mitigate the questions between, supplementing in a hatchery versus these wild stocks, and sort of eliminating any hatchery effects that may occur. But definitely we have programs throughout the basin that incorporate wild stocks into their hatchery programs to try to mitigate some of those issues. And that just sort of program dependent, species dependent, and it's not, I guess, widespread around the basin, but I guess.
Oh, sorry, we lost your audio for a second.
Oh, sorry. Yeah, sorry if I missed anything. I guess Shawn can probably fill in on some of the other species.
I think Rebekah covered it well for now. If there's other people that want to respond first, that's fine.
I was wondering and curious from my question for the panel is like, what is kind of like the minimum viable population, and how would you, I'd heard like people like in Katie's lab talking about like deleterious, deleterious alleles in the population, and like modeling that and seeing like, I mean, that's kind of what we're thinking about with chestnut, is we can match the adaptive allele frequencies with 100, 200, 300 individuals and across the range. But another thing is to consider is like predicting deleterious alleles, which maybe is a little on shaky ground, and then seeing like modeling, like simulating like how those will change in a kind of restoration setting. So I'm just curious from other people, like what are the criteria you're saying, like is your founder population for reintroduction?
Well, when we're talking about extremely endangered tortoises, the answer is as many individuals as you can get, which in some cases was, so the founding population of the Espanola species is 15 individuals, 12 females and 3 males, because that's all that was left. And so they were bred in captivity together for more than 50 years. So those individuals have hundreds of offspring. And so the idea is, I mean, you can't avoid how narrow that bottleneck is, but to make that be as short-lived as possible. So to really increase population size very rapidly. And that's been the approach, is when you really have very, very few founders, just to make that as short a bottleneck as possible, which with tortoises being very long-lived, lots of reproductive cycles, does give that opportunity.
A related but slightly different question for us is a lot of restoration practitioners were interested in how many corals they needed to start with for their nursery. Like, how can you capture allelic diversity? So less on the concern about deleterious alleles, because corals, at least in our group, were pretty much the point of saying, well, anything is better than nothing at this point, which convincing the regulators that that's the state that we're in is sometimes difficult, but that's a separate issue. But yeah, try to figure out, what's the minimum end for the strategy that we were recommending at the time. Basically found that with 3 coral colonies, you're capturing about 50% of the allelic diversity, which was, we thought, pretty good, particularly for small organizations that are wanting to get involved. You can start with a small number as long as you're sampling them from across a wide diversity of habitats. within a particular area. And so the guide was, if you think about your populations of nurseries, if you're trying to source them from different locations and maximize allelic diversity, given that you have restrictions on how many corals you can actually manage at a given time and keep track of and keep healthy, the recommendation for us was, in the Acropora cervicornis and Acropora palmata in the Caribbean, was 3 individuals per sort of environment that you're sourcing from, because usually you're sourcing from within 30 miles or so, or less. Now, how did we get to that number? I can't remember. Misha Matz did those calculations. I wasn't sure if he was going to be here today, but I think he shows up for these sometimes, so you can ask him. But it was a modeling approach, again, based on genome sequencing from an acroporid in the Indo-Pacific.
Great. Awesome. I think now, since we've had a lot of questions from me, we can have some questions from other members of the reading group who are here. So we've already got a couple in the chat, which I will just read off one of these to you guys. And if anyone else has questions, you can put them in the chat, or if we have time, I can just call on people near the end. But so Eric is asking, artificial breeding is sometimes motivated and funded principally by economic interests rather than more typical goals of conservation. Being kind of promoted in the economic interest rather than preserving biodiversity or preventing extinctions. Do any of the panelists feel that tension in their research efforts, and how do they resolve that tension?
With the chestnut example, there's interest from both public and private tree nurseries to plant these disease-resistant trees. And so our approach has been to collaborate. We want to keep it in the public sphere. So we're trying to get federal funding from some of this Infrastructure Act kind of stuff that we can then revamp some of the tree nurseries that are kind of defunct and bring in, like, hopefully some job development in Appalachia, which really needs it. And like but there's definitely some like private interests that are sort of working to like, I don't know, like protect it, like the intellectual property protections kind of get in the way, I think, and we've kind of kept it. The trees are not patented. We have a lot of people that are backyard growers that would just want to put this pollen on their, backyard chestnut or something. And so we're trying to, keep it so that the people that are been interested in it for a long time can just do it as a hobby sort of, but also bring in these partners that can do this at scale. And it's important to have the infrastructure and to make it cheap enough to where you can plant a seedling that's costs like $1 or less or something to produce. So I think like having, private entities work on this to make it more efficient is also important. So I think we're trying to do both public and private partnerships and not let anyone become like the master of this whole thing, like decentralized networks of people working on it.
I'll add, for a lot of systems that we're involved with, the goals largely are conservation focused, but there still is a lot of contentious issues around hatchery rearing of salmon and releasing them into populations where they could breed with wild populations. And so there's this dichotomy of hatchery versus wild. This is something that's common in our field of what is the best way to recover these source populations. And in many cases, hatcheries can be a tool that can be effective, but they can have some negative effects. So we need to think about how we balance the benefits and the risks of some of these supplementation efforts. And a lot of it comes back down to some of these conservation priorities, like we mentioned, of maintaining not only genetic diversity, but phenotypic diversity, as well as reducing effects of domestication selection on these populations. And some of it also depends on what the particular goals of a program are. If it's particularly a supportive breeding for a population that's about to be extirpated, we can follow those conservation practices pretty closely, as opposed to maybe there's another program that's intended to produce fish largely for harvest scenarios, whether it be for native tribes or for other fishers that are out there.
Cool. Yeah, that's great. Another question from our reading group, from Rebekah Jane here. So, she appreciated what John had to say specifically about interpreting coral traits, and would love to hear everyone's ideas on how to account for or deal with genetic by environment interactions and effects on the desirable traits that you're trying to breed for.
Do I have to jump in here again? This is a coral question. Thank you. Okay, so this is a GBI is a potentially big issue for corals, at least, but it's not entirely conclusive yet, depending on which system you're looking at, which is true. I mean, it's always dependent on which population you're looking at, right? But it could be a real problem. We have studies with different outcomes. Certainly seen examples where people have planted the same coral genotype at 10 different reefs, and you cannot predict which individual is going to perform the best at those different reefs. We've also had other studies where people have looked at basically finding the opposite, that you can sort of consistently, top performers-ish in like the top 10 percentile will tend to stay in the top 10. It really depends on the system. Do we have a really good solution for dealing with that? We definitely don't right now. And even attempts at artificial breeding in corals at the moment are just challenging in general. So getting that level of control in this system is definitely a problem. And there's a debate about whether it's worth trying to selectively breed for a particular trait, or just breed for diversity and cross your fingers and let natural selection do its thing. And that's sort of where we're falling on the spectrum at the moment for the system. The G by E and just how heritable some of these traits are is a major question when it comes to corals. Whether what the future conditions are actually going to be and which traits are actually going to help you with those conditions, it's hard to predict the future, right? And even knowing, if you're selecting for, say, heat tolerance, which we anticipate is going to be a bigger issue as the future comes, I don't think anyone doubts that. What are you selecting against at the same time? The same goes for when you're rearing them in a nursery. Whether you're doing that in the ocean or doing it on land in the ex situ nurseries, how much selection are we imposing? How much are we manipulating the diversity out in the wild when we are planting specific genotypes repeatedly in the same area? Are you swamping the potential future genetic diversity with those particular parents, right? We've actually run into a problem with that recently, where we realized, hey, look at all this successful coral restoration, and then, oh wait, all of these guys are siblings. Like, have we really accomplished our goal here? And so this is my non-answer for you. It's the problem that you're pointing out is a big one, at least for us. And if other people in other systems have better answers, that'd be cool. Or we're all equally screwed. I don't know.
I mean, the fact that you're seeing siblings do well, like that populations go through bottlenecks and then like rebound naturally. I mean, that's a curious thing. Like, we were worried about, we want to conserve 99% of the diversity, but like, is that necessary? I think it's like nice to like, it's like a lofty goal. But is it necessary? I think these conservation, case studies show us that maybe you can get by with something more limited. But yeah, it's interesting. But yeah, we have common gardens. We can plant, trees from the south and the north and the south and look at local adaptation. And we do see that is somewhat of an issue. And we're talking about trees with long lives. So like some of the things are like, hey, maybe it's like 100-year event, like some kind of weird climate, like extreme temperature thing, like in like a late frost or something that really matters. And we can't necessarily see that in the scale of, the decades that we're looking and planting these populations. So I'm just curious about those like really extreme events that might be the most relevant for long-term survival.
I'll also chime in that, the environment aspect of it is so varied and can't control for, and there's so many variables, in the coho system that we're trying to help support, this breeding of the stock to come back to this particular watershed. And they're doing it in big numbers. But the problem is they're only sort of reaching sort of where the dam is. And then all of the good spawning grounds are above this. And so they can get their way around it, but they're not actually getting up there. And that's one of the questions, they had us come in and look at is there sort of a genetic component of individuals that maybe can make it up to these good spawning grounds versus those that can't? And that's something we are trying to look at with the whole genome resequencing. But, there's other factors such as they have to navigate this really narrow and high gradient canyon. And I've seen pictures of it. The water just flies like crazy through there. And so it almost seems impossible that any fish can make it up that canyon, but we know that some of them do. But is it just a factor of maybe some years the water level is lower, so they can make it up? And there's nothing to do about it genetically. It's just, the flow of the water. Or it could be just body composition in general. We know the males have a easier time getting up that canyon than females. And that's something we don't quite yet understand. Maybe it's because the females are carrying so much extra, all the eggs, and sort of more of their resources are depleted. And that could go back to actually how much they're eating in the ocean. So I feel like there's so many factors that you have to take into account when you're thinking about those kind of G by E studies, because it's almost the easy part at this point is to figure out, okay, well, we can run some genome scans and see if there's any differences, but what other things, in the environment are going on that we have to take into account.
Great. Yeah, we had some other comments from Shawn and Rebekah Jane about about the value of common garden studies and the challenges of those studies too. And I think, well, we're getting up to 2 o'clock now. So I understand if people have to leave, but if we want to continue the discussion a little bit, I think maybe a good place to leave off would be, since we're kind of talking about some of the challenges that we're running into and some of the unknowns, are there any other research gaps that in your system, or more kind of generally in these kind of artificial breeding systems, research gaps that you've encountered, like your dream study of what you would, if you could, had, unlimited resources and you really wanted to answer one of these questions or address one of these gaps, what would you do in your system to really kind of improve the success of this breeding program?
One of the things that I'm really curious about is like, how did this quantitative type of resistance arise in evolutionary time, and what are the, you know, precise genetic, I mean, there might be a lot of routes to this, like there might be a lot of genes across the genome that contribute, and different trees have different genes. And I'm just wondering if, you know, pinpointing and understanding that to like that reductionist level in like the model systems, like it'd be amazing to see like there has been like some discussion about like, let's say, can you convert an elephant to a woolly mammoth or something, you know, this, you've probably heard about that project, and like they're going like brick by brick trying to change the elephant genome to the woolly mammoth genome, but like understanding quantitative traits to where you could do some kind of genome editing, or like we could take a swath of genes from Chinese chestnut and insert them in one place in the American chestnut genome, just kind of we don't have like that kind of baseline level of information to be able to do that.
Yeah, I'll add to that, you know, and saying doing these genome level scans, you can find these really nice peaks, you know, that maybe highlighted a particular gene that may be of interest to you, but that always nags at me is like, what does that gene do? And how do we figure out, you know, is this really something that maybe affects, you know, the phenotypic trait? And getting at that in non-model species is just so difficult. And it seems like I don't know when exactly that is gonna sort of start to happen. But it'd be great to have that sort of level of what we are finding is that actually have an effect on the phenotype would be nice.
And I think a big gap that still remains for us is on the symbiont side, right? And so you asked about my fantasy of what I would do with all this money if we could. And it would be to get at the symbiont at the same time that we're getting at the host, because we don't really have any management plans or strategies in place for symbiont diversity. And we don't have a really good understanding of symbiont population genetics. We do get that it tends to be a bit more structured than the hosts. But it just tends to be understudied. And we have a lot fewer symbiont genomes than we do coral genomes. And those symbiont genomes, they're dinoflagellates, and they're the worst. They're multiple gigabases, they're highly repetitive, there are all sorts of weird quirks that dinoflagellates have. And so trying to develop genomic resources for them, which is what I've been spending the last couple of years doing, is hard. And then you have it, and then like what do you do with it? Because it's even hard to interpret the data that you get back. You know, is it whole genome duplication, or is it sequential duplication, segmented duplication? You know, are we getting these weird number of reads because only portions of it are duplicated? You know, why is our coverage spiking for this gene and not that one? We only have 2 quote unquote chromosome level assemblies at this time. And there's a question as to how representative they are. And honestly, we have like 1 genome per species. We don't even have multiple genomes of multiple strains within a species at this point. We're working on it, but it's just slow to do because they're difficult. If we had genomic resources for them, if we could knock out genes, obviously that'd be great. But even if we didn't have that, if we could have do a better job of doing shallow sequencing, even on the symbionts that we do have, I'd love to look more at that. And we're hopefully going to do a little bit of it because that SNP chip has some symbiont markers on it as well. And we're doing this project in Florida that involves us doing heat tolerance assays, phenotyping a bunch of colonies, and doing resequencing. And we'll definitely pick up symbiont reads, but it's not the focus. So, and there's like a 10-to-1 cell ratio, and then you extract everything, you get different components because the symbionts are hardier. It's not focused on the symbionts. I prefer to be able to look at that and then develop some sort of management plan for symbionts as well. And having a good link between symbiont phenotype and host phenotype would also be lovely, because that would also inform the management plan. But we're just not there yet. Lots of unknowns with these guys, but the symbiont is really important. And I'm just talking about the dinoflagellates. The other components of the microbiome are also important. And relatively understudied. So big gap there.
Yeah, I think my sort of dream is to have some of the genomic resources that they have for other organisms. So in tortoises, you cannot do experiments of any kind. It would be lovely to have a chromosomal reference genome we don't. It would be great to have a recombination map that people that study turtles have to use the chicken recombination map, because that is the closest thing. And it's just trying to do local sort of ancestry reconstructions and things. It's just, we don't have the tools, and it would be really great to have them, which, I mean, yeah, maybe this will all resolve in a number of years, but where we're at right now is it's really holding us back.
Oh boy, yeah. Well, yeah, okay. So that's those are all kind of, yeah, hard problems to solve, but yeah, I'm hopeful and glad you guys are all doing this work and advancing the field. So yeah, it's around 10 minutes after 2 now. I think unless anyone else has any questions, I'd like to just thank everyone for coming and participating, and yeah, I will put this recording up on YouTube for anyone who wants it. If everyone is okay with that, I think everyone is fine with that. Cool. Okay. Yeah. So this will be up on YouTube. It'll be a good resource for people who are interested in all of these issues. And thanks to all of our panelists and all of our attendees. And yeah, talk to you later. Bye all.